Thank you, Neelam. Thank you, the sponsors, and special thanks to Dr. Krinal, and I'm here today just because of him. I just thought it becomes very monotonous throughout the day as you discuss vaccines, antibiotics, infections, mimics. So I just thought, let me keep the topic a little bit different. And I think we now need to understand that we are in a microbial world, and I'm just a speck in a microbial world, and we are run by microbes. So if my body temperature is 99, 70% of it comes from the microbes. If I've just finished a very good meal in the Vegas hotel, 70% of the digestion is by the microbes. If I start feeling attracted towards the moderators, it's the volatile organic acids generated by the microbes. So there is a role for the microbe in almost everything that we do. And the basic thing that I'm going to stress upon is we have messed up with our microbial system. I really want you to go through these two articles. The first one is the husband-wife combination, Sonnenberg. I think you must have read about a lot of their articles on the human microbiome as well as what is happening to the ancestral microbiome. And the second one is how to restore this new entity. Just like we have a sleep debt, we have something called as the microbial debt. And most of the diseases that we see today, the non-communicable chronic diseases, are linked to microbial insufficiency syndrome. So as we enter the gut, we do realize that they are all there. And we know they are there because we have all the new generation techniques by which we know they are all there, but we are not able to culture them. And now we have the artificial intestines like the chyme. So you could very well grow some of the fastidious organisms and know that there is much more than what meets the eye. And you need to understand that majority of them are symbiotic or beneficial, and it's a very small percentage which are pathobions. This is what really stimulated me. I went through this book. This is way back in 2014, and it was written by Martin Blaser. I don't know how many of you all know about Martin Blaser. He's one of the 3 editors of the Mandel's textbook of infectious disease. Fegin and Cherry is one book that we all go through. Mandel's textbook is the other one. And he is not only a professor of medicine, but also a professor of microbiology. An amazing book. And he links the modern plague of non-communicable chronic diseases like obesity, like allergy, like the chronic inflammatory disorders, allergy, to the missing microbes, which is probably because we started getting very excited with antibiotics and we thought that's the end of the infectious era. But unfortunately, now we do know that you can't outrun your shadow. And we are actually just a speck in a microbial world. So let's talk a little bit about the ancestral microbiome that our ancestors carried. And how did we study this? The coprolites, the fossilized feces, and from the mummified bodies, we did study the microbiome. And what we did find is diversity, phenomenal diversity, numbers, great numbers. And the carbohydrate accessible enzymes that these bacteria processed was phenomenal. We don't have them any longer. And they were never dominant because it was diverse. So in this room, if I had an orthopedic surgeon, a dermatologist, and all the specialties in this room, it would have been much better than just having all the pediatricians together. They would have contributed much more. So diversity rules in everything. So we are losing on diversity, we are losing on numbers and symbionts. So what is actually happening is, at the level of the intestine, we have something called as controlled inflammatory tone. And this is going out of control. And this is happening because we are missing out on the beneficial microbes. Also the diet, look at the diet. The male hunters would go around hunting, the females would pick up the tubers, the plantains, the roots, the vegetables. So it used to be a mixed diet, and there was never a surplus of diet. You could have scarcity. So there were times when there was nothing to eat. So there was no dominant organism that would take over. And we used to call this taxa in the ancestral flora as the vanished taxa. Volatile and no longer seen in the industrialized society. And most of them were the motile ones. So we had the Prevotella, we had the Succinivibrio and the spirochetes. And there were plenty. And they were not constant. And the diversity was phenomenal. And the net result is, what you see here is, you have a genome that is very well adapted to the gut microbial community, and the incidence of communicable chronic disorders which are non-communicable was not seen. Now what has happened in the present era is surplus of diet, ultra processed. So you have something like hydrogenated oil, hydrolyzed proteins, modified starch. You start giving formula feeds. Even one formula feed can change the flora. The breast milk has something called as HMOs, human milk oligosaccharides. The food for the bifidobacterium which supplies acetate. There are cross feeders and you get butyrate, and these short chain fatty acids are needed for barrier integrity, and not just barrier integrity, for colonocyte function and strength. So if the barrier is maintained, nothing gets in. So you have no autoimmunity, you have no allergy. When you lose the barrier, there is all sort of mischief. You have an immune system on the other side which is totally confused, and you are on the edge. So you have an immunity on the edge, and you have the microbiota on the other side also on the edge. But the microbiome can adjust very quickly and very fast. They can mutate. Conjugation. They can pick up DNA from the surroundings, and they are extremely promiscuous. They can transfer from one to another, and they can adapt. And they can adapt very fast. So what is happening is, with the present day diet, you are not producing or you are not providing enough fodder for the symbionts. Second is distance. There is no such thing as distance in today's era. Travel. You have the aeroplane there. Traveling has become so simple. And that's why the exchange, the food, the disease, everything is so convenient. So these are two things that contribute to a condition called dysbiosis. So what is dysbiosis? When you are not in synergy with the microbiota that you carry. Now if you really see that in the present era we are seeing so many new diseases, and there are so many new drugs. You have antibiotics, you have antacids, you have antitussives, you have antipyretics. Let's take each one of them. If you take the antibiotic, it's a stress. It's not just going to get rid of the organism that you're targeting, but it has a collateral effect. If you take an antacid, you need to understand that the first line of defense is your acidity. The first line for stimulating pepsinogen into pepsin for protein digestion is the pH. So you're doing a mistake by taking a PPI, an H2 blocker, or an antacid. Antitussive. All those antihistamines which are first generation are anticholinergic. Motility is affected. So you promote growth of the wrong organisms. Antipyretics, antiprostaglandin. So the epithelial regeneration again is affected. There is a barrier defect. So every prescription is a prescription for disaster. So don't pen any drug unless and until it's needed, and always mention SOS when you feel that it needs to be given only if needed. So the net result is you start getting organisms which are resistant, which are resilient, because they are smart. They switch on their virulence genes, they switch on their resistance component, and you are creating a resistome in the gut. And some of them do very well by eating the mucus, the glycans of the mucus. Some are beneficial, like the Akkermansia muciniphilia, because they eat up the glycans, they generate a lot of butyrates which are beneficial. The pro-inflammatory microbiota can go right up to the epithelial structure and bring about a major inflammatory change. Like I told you, the immunity is on the edge. So when you get your microbes from the mother, how many of us do know that the neonate in utero is no longer sterile? There is a transfer of metabolites, there is a transfer of microorganisms. And these microorganisms are known to develop the immunity. So when you are born, you are already conditioned to take in the mother's microbiota. But if it is a cesarean section, you're delivered in a hospital, you pick up the hospital microbes. And that is when the confusion starts and the trouble begins. So the net result again, what happens? You have a non-adaptive system. The microbes have adapted pretty well, but they have kept their virulence gene switched on, the resistome switched on, and there is a high incidence of non-communicable chronic disorders and microbial depth. See the difference. Vanished taxa, rare. Akkermansia mucophilia, abundant. The bacteroids are high in the industrialized population. The diversity is pretty low. The carbohydrate accessible enzymes are low, and you don't find the seasonal dynamics that we used to find in traditional populations. So you're allowing certain pathobionts to remain there. And they have a system called as quorum sensing, by which they can increase their number in just a jiffy. So you have to understand this, and that is why we are going through so many epidemics. So this takes you to something called as the hologenome theory of evolution. We all know about the Darwinian theory. The Darwinian theory is survival of the fittest. The hologenome theory or the holobiont theory says that every structure, like if you take the human beings, it's a summation of the human cells and the microbes. And if you take the ratio, for every human cell we have 1.3 microbe. See, here also the microbes are much more. And if you take the genome of the human genome, if you consider, it's almost 99% microbial genome. So they are all there with a purpose. And if there is a cohesiveness or synergy between the two, you do very well. So that's what the hologenome theory states. So now we are in a state wherein the microbial part is well adapted, but the genome part, the host genome, is on the edge. And this is not a good combination because the microbes develop pretty fast. The host genotype takes a long time to evolve. So this comes under a new classification of medicine called as evolutionary medicine. So in the present era, we have microbial depth, and we call this as microbial insufficiency state. And because of this maladaptation, what we are seeing is, we are seeing antibiotic resistance to the fore. We are seeing developmental diseases. My genome knows to accumulate the thrifty gene. So when there is an excess of food, you're bound to get obesity. Midlife illnesses. What is midlife illnesses? We see a lot of stones nowadays, renal stones. Because we used to carry a bacteria called oxalobacter, which used to take care of oxalates. They are missing. So the incidence of stones has gone up. There is a barrier defect. The barrier is maintained by the ancestral or the good microbiota, which is missing. So if the barrier is missing, you are more exposed to the wrong ones, and you have autoimmunity, you have allergy, you have translocation and various diseases. And the last is epidemics. An MRSA in the institution, a VRE. This is because they are all kept under control by the symbionts. So if I have a corynebacterium sitting in my nose, it keeps the MRSA under control. If I have the right organisms in my gut, I don't generate a VRE. So the maladaptation is responsible for all this problem. So the last 7 minutes would be on live microbial therapeutics, which is very close to my heart. And I'm just thinking of a therapeutic intervention by which I could regain what I lost over the years, which is going to be pretty difficult. And this promise is there because fecal transplant came into play. So we came across a condition, I think the gastroenterologist would be able to discuss this better. We had a number of C. difficile cases that came up, and they were recurrent C. difficile, and fecal transplant brought about a dramatic change in the management of the recurrent C. difficile, because we used to use antibiotics there. So C. difficile was predisposed by use of antibiotics. We used to use metronidazole, we used to use vancomycin, fidaxomicin, nitazoxanide. It's a big list. You're using an antibiotic again, recurrence is pretty common. And any of these drugs can give rise to a recurrence. So we started using an antibiotic a microbial cocktail and fecal transplant, and we started getting dramatic results. So that's what brought us into live microbial therapeutics. The problem with live microbial therapeutics is you are using a microbe to treat a condition. So when you are using a microbe to treat a condition, you are scared. A microbe can mutate. So a microbe could be different outside. The moment it enters my oral cavity, it changes. It enters my gut, stressful, it can change. By the time it reaches its colon, it could be a different microbe. So it's very essential that when you select a microbe to treat a condition, you need to know its genome stability. So that's why we have a repository number, ATCC. Every probiotic which is used as a microbial therapeutic is kept in a repository with a number and studied monthly, every 6 months for resistance. Then we also look at the antibiogram. The antibiogram is what antibiotics can finish off this microbe in case it translocates. Because if you are using a microbe, it can translocate. So we have certain conditions wherein we don't use a live microbial therapeutic. For example, burns, or where translocation is possible, an immunodeficient kid. Preterms, it's a risky proposition. So you have to be careful when you have central lines. So if you have the antibiogram and you have had a 1:1 talk with your parent and you are using it for the beneficial effect and it gets in and your antibiogram is there, you can treat it. This is something which excited me very much. This is a 2024 article in the International Review Journal. It's an in vitro study where they compared the main probiotics in the market. You have the Ruteri, which is used for colic, which is there in infant formula. You have Saccharomyces boulardii, the fungus, a great probiotic, a Lactobacillus GG, the maximum publicity that it has had. Bacillus species which have now come to the fore. And I am talking on a Sanofi platform, but I am talking on facts and factuals. Now, when you look at this table, which is a summary of what has been put in this vitro study, for a probiotic to work, it needs to get in. If it does not survive, it's not going to work. Second, it has to get attached to the epithelial side. So it has, as you move from the small intestine to the large intestine, the mucus layer goes on increasing in size. Once it attaches in aerobic, anaerobic conditions, it starts producing metabolites called short chain fatty acids, acetate, propionate, butyrate, and they can be converted from one form to another by various bacteria in the gut. Acetate, very useful for lipid metabolism, propionate, glucose, butyrates for stability of the colonocytes and barrier integrity and distal effects. So these molecules work not only locally but also distally. Then they also bring about an alteration in the redox potential of the gut. So by producing certain molecules that take care of oxidizing agents in the gut or an oxidizing insult to the gut. And you can produce enzymes like beta-galactosidase, which is nothing but lactase. So if you want to avoid a post-gastroenteritis syndrome, which is pretty common after a viral disorder, you start off with a probiotic right in the beginning and it has the properties, it would be beneficial. And when you look at this table, one thing which fits in all the blocks is Bacillus clausii from Enterogermina. You need to understand there are a lot of clausii in the market. You have an UBCC, you have biosimilars. They are all not the same. You need a repository number, you need a genome to be decoded, you need to know the stability, and you need to know in vitro and in vivo if possible. So if I'm going to be using a clausii for an antibiotic associated diarrhea, I know it produces proteases that can destroy the toxin A, toxin B of Clostridium difficile. So I need to know all these properties. From this you can very well see that the Bacillus species is coming to the fore because India is a country where the biggest problem is temperature. And I'm wearing a coat and I'm standing here in a black dress because the microbial matter is called the dark matter. But this is not suitable for the climate. Heat is absorbed by black things. But I'm going to remove this as soon as I move out. But what is important out here, what I'm trying to say is, The bacillus species are in spore forms. The only mistake that we do when we prescribe this, the market went up just by the word of the mouth from almost 0 to 200 crores today. And it's in a spore form, extremely stable. And all that we need to tell our parents is to shake the vial before they give it, which we forget to do in our busy OPDs. If you take a lactobacillus or a bifido, they are very thermolabile. And a lot of properties are on their pili, so they require a specialized type of formulation called the pigylation for maintenance. Fine? So you need to understand why the bacillus spores are coming to the fore. And I just want you to look at stool banking, designer probiotics, and autologous poop transplant. I think it's not far before we start doing stool banking, much better than stem cell banking. First 2 years, start storing your stool cryo in the freezer, and it could be useful later on because you know everything starts off from the gut. Hippocrates said this long time back. All diseases begin in the gut. We need evidence. Our grandmothers also knew this. So what is important now is to take care of your microbiota, restrict your drugs in your prescription, use an antibiotic which is narrow spectrum, know the collateral effects, and that's something which is very essential. So they are all there, but they are there with a purpose. And most of them are symbionts. A negligible amount are pathobionts. And we were only told about prions, and now we have a new microbe there which is called the obelisk. I don't know how many of you all know about it. This is an RNA particle. So the new thing there is the RNA particle called obelisk, and we don't know what they are doing out there, and they are in huge numbers. We have still not dissected out the fungiome and the viriome, the bacteriophages which can alter your microbiota. So we need to remember that I am just a speck in a microbial world. I am full of microbes inside, outside, and surrounding me. And they are all there with a purpose. And probably life began with a microbe, and I'm alive here because of a microbe. And when I perish, the microbes are going to have the last laugh. So I think it's time for us to respect the microbes in our body. Thank you.